In this video, we're going to talk about osmoregulation. So let's start by doing a review of what osmosis is. So osmosis is a type of transport across the cell membrane, and specifically osmosis is the movement of water from an area of lower solute concentration to an area, to an area of higher solute concentration. And remember that when we're talking about solutions, so a solution is just an amount of solute dissolved in a solvent. And in this case, the solvent that we're talking about is water. I have a whole other video that goes over osmosis and how it occurs, so I'm not going to go into those details here, but I will post the osmosis video if you want to go back and review or learn more about that concept. Now, anytime there is an imbalance of solutes on either side of a membrane, osmosis could occur. And this imbalance of solutes causes something called osmotic pressure. So you can see in this hypertonic solution, there is osmotic pressure. And that's because the concentration of the fluid outside the cell, which we will just call the extracellular fluid, is going to be more concentrated than the solution inside the cells, which is the intracellular fluid, also known as the cytosol or cytoplasm. A hypotonic solution also has osmotic pressure, but in the opposite direction, because in this case, the extracellular fluid is less concentrated than the intracellular fluid. An isotonic solution does not have osmotic pressure. And that's because the extracellular fluid is equal in concentration to the intracellular fluid. On the next slide, we're going to talk about osmoregulation, but I want to talk about this picture in relation to osmoregulation, because this picture shows you why osmoregulation is important. So in these pictures, you are seeing red blood cells surrounded by a liquid medium, which in this case is blood plasma. And you can see that when there's not osmoregulation, we end up with situations that are not healthy or not good for the cells. So in a hypertonic solution, this is bad. So cells in a hypertonic solution will lose water and become shriveled, meaning that they can no longer perform their duties. Cells in a hypotonic solution will take on water, causing them to swell up and potentially burst, which is also not a good thing. But osmoregulation happens when cells are isotonic with their environment. So this is the isotonic solution is what we would call homeostasis. And osmoregulation is how we maintain that homeostasis. So as I mentioned, osmoregulation is a form of homeostasis. And to review, homeostasis is just that steady state of internal conditions that can adjust based on changes to the environment. So you're constantly taking in water and electrolytes, but you're also constantly losing water and molecules such as electrolytes. And so your environment is constantly changing, and your body and your cells have to be able to maintain that homeostasis in order to keep your body functioning and keep you alive. So osmoregulation is a type of homeostasis that more specifically is the process of maintaining salt and water balance across membranes within the body's fluids. And the body's fluids are composed of water, but also electrolytes and non-electrolytes. And we're going to talk more about the difference between an electrolyte and a non-electrolyte on the next slide. So first of all, let's talk about electrolytes. So an electrolyte is a solute that dissociates into ions when dissolved in water. Electrolytes are ionic compounds. And so ionic is just a form of chemical bond in which one atom gives an electron, one or more electrons, to another atom, and then they are attracted to each other because they now have opposite charges. Ionically compounded molecules are also just known as salts. So the salt that you're probably most familiar with is sodium chloride, or NaCl. And this is more commonly referred to as table salt. So this would be the salt that you would have in your salt shaker that you would put on your food to make it taste better. And when sodium chloride is added to water, it dissociates into sodium ions and chloride ions. And so sodium and chloride are the electrolytes, and they are ions. And notice that they have different charges. So sodium is a cation, which is a positively charged ion, and chloride is an anion, which is a negatively charged ion. But table salt is not the only example of a salt. And so things like hydrochloric acid are also considered a salt. So hydrochloric acid, when dissolved in water or added to water, it dissociates into hydrogen ions and chloride ions. Magnesium chloride dissociates into magnesium ions and chloride ions. Sodium hydroxide dissociates into sodium ions and hydroxide ions. And calcium carbonate dissociates into calcium ions and hydrogen carbonate ions. And in this case, that hydrogen in hydrogen carbonate comes from the water molecule. Non-electrolytes are molecules that do not dissociate into ions when dissolved in water. Some examples of non-electrolytes would be things like sugars and many other organic compounds. So let's talk about why osmoregulation is needed. So think about humans, for example, but this applies to other animals as well, where electrolytes and water and other molecules and compounds as well are constantly being added to or removed from the body. So think about when you eat something, you're taking these molecules and water and you're putting it into your body, and it goes through your digestive system and gets absorbed into your bloodstream. You are also constantly losing electrolytes and water through different body fluids such as urine, feces, sweat, saliva, etc., etc. Now, this whole time, you have cells in your body and remember that these cells have an intracellular fluid to them, so a fluid inside the cells. And surrounding these cells, there is a liquid environment. And this liquid environment is the extracellular fluid. And remember that if the concentration of the extracellular fluid becomes hypertonic or hypotonic to the intracellular fluid, then the cells are going to be damaged and they're not going to be able to work properly. So they might shrivel up if they're in a hypertonic environment, or they might swell up and burst if they're in a hypotonic environment. So in animals that require osmoregulation, there are structures and systems in place that work to maintain an isotonic extracellular fluid. So you think about the cells need to stay the same. So they're going to have a certain intracellular concentration that really should not change. It's the extracellular fluid that is constantly changing with regards to how much water and salt is present. And so, for example, in humans, the kidneys work to get rid of excess salt and or water or retain salt and water in order to maintain an isotonic extracellular fluid so that our cells can work properly. So we have to keep our cells happy so that they perform their functions. And to be happy, they need an isotonic environment to survive in. So let's take a closer look specifically at mammals. And so mammals have these kind of three different fluid or liquid environments inside their bodies. And the electrolytes and other molecules in water are constantly being moved between these different compartments. So let's talk about these different compartments. So first of all, I'm going to draw a blood vessel here. And inside this blood vessel, you have a lot of water. And so I'm just going to draw some little fake water waves here. And that liquid or that watery part of the blood is known as the plasma. And it's not just water, there are other molecules associated with it as well, but it is this mostly watery substance that's moving through your blood. Now you do have red blood cells moving in that plasma. You also have white blood cells and proteins and other things that are also in there, but I'm just going to focus on the red blood cells for now. Now, your blood, the purpose of blood is to transport different molecules to the cell and then remove other potentially negative molecules away from cells. And so these molecules can diffuse out of the blood and into the body cells. So I'm going to draw the body cells here in green. And inside these cells, there is a liquid environment known as the intracellular fluid. And the intracellular fluid is also known as the cytosol or the cytoplasm. And then there's also liquid outside the cells. So there's going to be some kind of here in between the blood vessel and the body cells. There might even be some cells that are separated from each other. And so there might be some extracellular fluid just kind of outside the cells here. And this extracellular fluid is sometimes also called tissue fluid or interstitial fluid. And if we look at the blood itself, the blood cells also have their own intracellular fluid, and the plasma kind of acts as the extracellular fluid of those cells as well. But for the sake of this, we're going to refer to the plasma more specifically as plasma, and then the extracellular fluid is going to be this fluid that is outside of the blood vessels, but also outside of the other body cells. So you can imagine that when you are, let's say you're eating, and let's say you take in some chips or something that have some salt. And so that salt is going to get, or those chips are going to get broken down by your digestive system and ultimately be transported into the blood. So in the blood, you're going to have some sodium and chloride ions that are floating around, amongst other things. And so now your plasma has become saltier or more concentrated. And so the fluids have to move in order to help compensate for that. So the interesting thing is that in some cases, the solute, so in this case, the sodium is the solute. as is the chloride. So the solute can potentially move between areas, but the water can also move between areas. So in order for the body to maintain this isotonic extracellular fluid, because remember from the previous slide, the extracellular fluid needs to be isotonic to the intracellular fluid. And so to do that, the blood plasma can actually work as a way to either deliver salts and water to the extracellular fluid, or it can take salt and water away from the extracellular fluid. So remember that this plasma, its main function is really transport. And one kind of key area that this blood gets transported to are the kidneys. And then there are structures in the kidneys that can control how much salt or electrolytes and how much water is either kept in the body or excreted out of the body. So the kidneys are really the organs responsible for osmoregulation. So your kidneys help to keep the extracellular fluid isotonic to the intracellular fluid. And the movement of these solutes and water are transported around the body from the digestive system to the kidneys and other places, but to the kidneys, at the kidneys, the materials are filtered, and the kidneys can actually adjust how much salt and water is retained or excreted in order to adjust the tonicity of the extracellular fluid to make it isotonic to the intracellular fluid. And we'll talk about how the kidneys do that in a different video. So electrolytes in water can cross cell membranes, but they require different processes to do so. So first, let's focus on the electrolytes. And remember, the electrolytes are going to be those ions. So things like sodium, fluoride, potassium, calcium, etc., etc. Now, because of this charge, they cannot cross the phospholipid bilayer of the cell membrane by themselves. So let's draw a cell membrane here. Let's pretend like this green circle is the cell membrane. Remember, the cell membrane is made up of a phospholipid bilayer. So you've got these phospholipids, and their tails kind of point into each other. And so ions are not able to cross the phospholipid bilayer. So if they try to get into the cell, they're going to bounce off, or if they're inside the cell and they try to get out, they're not going to be able to do so unless they have some help. So ions can cross the cell membrane in the form of facilitated diffusion or active transport. So first, let's take a look at facilitated diffusion. Facilitated diffusion is a form of passive transport, which means no ATP or energy is required for those substances to cross the cell membrane. So facilitated means help. To facilitate something, you help something. So the help comes in the form of a transport protein. And these transport proteins are required, and they are called either channels or carriers. In facilitated diffusion, the solute can only move down or with the concentration gradient. And what this means is that the solute is going to move from an area of high concentration to an area of lower concentration. So let's say we put our little channel carrier in there. In this case, this molecule would diffuse into the cell because there is a higher concentration of it outside the cell and a lower concentration of it inside the cell. In active transport, it is active. It's right there in the name. And active just means that ATP is required. And like facilitated diffusion, a transport protein is also required. And in this case, the transport protein is usually called a pump. And in active transport, the solute is going to move up or against the concentration gradient. So in this example here, if we were to put a different a pump here, so this is our protein pump, and let's say we've got a high concentration of whatever this molecule is outside the cell, and a lower concentration inside. In this case, the solute or the electrolyte, the ion, is going to get transported from the low concentration to the higher concentration area. So these forms of transport, the facilitated diffusion and active transport, can actually be used as a way for cells to control when and how much of an ion or an electrolyte is going to be able to cross the cell membrane. So with active transport, remember that you have to have that transport protein and ATP in order for it to occur. In facilitated diffusion, you have to have the transport protein. Sometimes those transport proteins are also gated. And gated just means that they can be open and closed depending on different situations. Water, however, differs from electrolytes in that it can be transported across the cell membrane via osmosis. Now, osmosis is a form of passive transport, so no ATP needed. It can also cross through the phospholipid bilayer on its own. So technically, it also does not need a transport protein. However, there are transport proteins called aquaporins. And so this is a transport protein specific to water. And even though osmosis can happen without aquaporins, if these aquaporins are present, then osmosis can happen a lot faster. So technically, water could cross the phospholipid bilayer, but if you had these aquaporins, the water could go in and out of the cell much faster. Now, there's a saying that water follows salt. And what this means is that in osmosis, remember, water is going to be moving to the more concentrated area or the saltier area because if the salt is not able to move in order to reach equilibrium, the water might move in order to try to equal out the concentrations on either side of the cell. So cells that might have a lot of pumps to actively transport ions across cell membranes are also going to be able to essentially create osmotic pressure to essentially suck water into different areas. And we're going to talk about the kidneys and more specifically the nephrons, and we're going to talk about how the active transport of salts like sodium can lead to the reabsorption or kind of the passive following of water. I want to take a moment here and talk about the difference between osmoregulators and osmoconformers. So far, the majority of what I have talked about in this video has to do with osmoregulation. And so osmoregulators are animals that are able to regulate their osmotic pressure in order to maintain that isotonic environment, that isotonic extracellular environment, to keep their cells kind of happy and healthy. So osmoregulators are able to live in a variety of environments. So some examples include things like mammals and reptiles and birds, and even some fishes. So for example, salmon are able to live in freshwater and saltwater. And so they have certain mechanisms and processes in place that allow them to adjust the osmolarity of the fluid inside their bodies by either taking in salt and water or releasing salt and water in order to keep their body fluids isotonic with their intracellular fluids. Now, osmoconformers are not able to regulate the osmolarity of their body fluids. And so examples of osmoconformers include a lot of marine invertebrates, and many fishes. So for example, for most fish, if you take let's say you go to the ocean and you catch a saltwater fish, you can't take it home and put it in a freshwater aquarium because it will die because it's not able to regulate its body fluids. And so if you put it into a freshwater aquarium, that water is going to rush into the cells of the organism, and those cells are going to swell and burst. Same thing with taking a freshwater fish and trying to put it in a saltwater environment. The fish is not able to deal with that extra salt. It's not able to excrete that salt out of its body. And so the salt is going to draw water out of the fish's body, and the fish is essentially going to become dehydrated and die. So not all organisms need to do osmoregulation, and not all of them do. But one kind of benefit of osmoregulation is the ability to live in a wider variety of environments.